The current scenario in ART training across the world is of observation only, which although gives you a fair idea of how the procedure is being done, but does not give you the confidence of performing it independently.
I just finished a visit to Indira IVF group in Udaipur, India, and I'm totally impressed by several things. But one of the things that I'm most impressed of is the way they just prepare themselves for the future and how they're expanding.
I'm here this afternoon visiting the Indira IVF Center in Udaipur. It has one of the most cutting-edge facilities that I have seen, including the perfect training for physicians and for embryologists. And I really think this is the future of how to train superb doctors and superb embryologists.
Embryology is confined not only to collection of gametes or making embryos, but it is about making euploid embryos to achieve a higher pregnancy rates. Embryology skills, lab culture conditions, gamete handling, and optimal environment are desired and necessary to achieve such results.
Yeah, Preta. So, okay, it is just a formal introduction with for the David, and just a minute. Although actually I'm not speaking it just for as the sake of saying that, but yes, obviously David, he really doesn't require any introduction, but since it's it's a formal way of always greeting, and I would really want to introduce David. David Mortimer, yes, as we all know that he's currently scientific advisor to Mellowwood Medical Canada, Planner UK, Hamilton Thorne USA, and as well as with several ART centers internationally. And he's also an advisor to WHO in preparing the third edition of their laboratory manual. He's the co-founder of ASHRAE Basic Semen Analysis course in 1994, president of Canadian Fertility and Andrology Society 2009-10, and associate editor of Human Reproduction Update 2011 to 14. So he has developed new type of sequential media, which we all know by the name of Cook culture system. And he's convener for 11 international consensus meetings, and that have been published in leading peer-reviewed journals, including Istanbul Consensus and whatnot. So here today we have David, and he'll be speaking on the topic, don't let your eggs get cold. So the importance of temperature control during oocyte retrieval and handling. And without wasting any time, over to you, David.
Thank you very much, Ritesh. So it's a great pleasure and an honor to have been invited by the Indira Fertility Academy to speak to you today. And my topic, as Rich suggested, is don't let your eggs get cold. Some of you may have seen at least some of these slides before, but this is a topic which is one of the biggest issues we find when we visit labs, audit labs, and it's something that I don't think we can ever repeat enough, because people just don't seem to focus on the most crucial raw material that comes into the lab, which is the oocytes. So we're going to talk about this today and hopefully have some interesting Q&A at the end of this. Okay, now just going to make sure that's it, good. So let's start by a slide we used many, many times, the IVF Lab Prime Directive. Biology rules. The entire process of IVF is governed by the biology of the gametes and the embryos. It's up to us in the laboratory to provide optimal conditions for the gametes and the embryos. We have to protect the gametes and embryos from adverse external factors and protect them from physiological stress. In other words, not cause physiological stress. Embryos are highly adaptable for sure, but adaptation costs energy, and that means it's metabolic stress. So yes, they can adapt, but we'd rather they didn't have to. We know that cellular stress can alter gene expression and or gene regulation, for example, expression of heat shock proteins or imprinting patterns. We know that suboptimal embryo culture can also affect the health and behavior resulting adults in experimental work, obviously from mice. And these biochemical and biophysical requirements govern all of the ART lab procedures, as well as the design and construction of the lab, the HVAC system, the engineering of the lab equipment, and of course the culture materials that we use. ART lab processes must support and respect the natural process, including its timeline. So we're there to do what we do, but totally within what nature expects. So we did a consensus meeting about 2019, and these are just a few shots of the people working way around the table. If you notice, Shannon, everyone is busy sitting writing because she's the recorder for these meetings. But the consensus that came out of this one about IVF culture conditions was actually from Don Riga, and he said there's only one thing that's truly important in an IVF lab: everything. So that's what we need to remember. Everything we do affects the outcome. So I'm going to focus on a few areas now that relate to egg retrieval and egg handling, and using statements from the consensus guidelines. So workstations, whatever the workstation is, it should be designed and operated to maintain the oocytes and embryos as close to 37 degrees C as possible while minimizing pH shifts. When you're working in dishes without oil, every possible precaution must be taken to minimize evaporative cooling. And the travel distance between workstations and incubators should be as short as possible. So this is an example of a lab design we did quite a number of years ago, because we've still got the old M-cell workstations. But in this process, you can see that we've arranged the lab, which is 2 procedure rooms, with functional zones where we're doing ICSI and biopsies over here. We're doing the embryo fertilization and culture here, and then we've got embryos in the latter part of culture before they go back into the procedure for transfer. And the basic process is that equilibrated culture dishes move from this equilibration incubator here into the workstations. Follicular aspirates come from the procedure room to the workstation. An egg dish, which is the dish that will be holding the oocytes as we find them, moves from an equilibration incubator, in this case it's a benchtop, next to the workstation. And at the end of the procedure, the eggs are put into culture via a short distance, as short as possible, within the confines of creating the laboratory. So we design the labs to minimize not just the risk of paths crossing, so to avoid people having accidents and dropping dishes, but to minimize the movement required by the staff doing their procedures, which also reduces risk of collisions, but also to make sure the transfers are as close as possible or as short as possible, so we can actually maintain the equilibration conditions in the dishes and handle them carefully and appropriately. In terms of handling practices, the consensus guidelines say that every attempt should be made to maintain the temperature of the oocytes and the embryos at 37 degrees C throughout every step of the process. Care should be taken to minimize heat loss in pipettes and oocyte collection needle sets. And if a metal carrier is used when moving dishes between the incubator and workstation, the choice of device should be based on results from simulated procedures that have assessed the temperature of the dish contents. So you do dummy procedures or whatever, but with thermocouples in place, so you know what's happening to temperature control. Your scientists do experiments, measure things, obviously not with oocytes in the dishes, but you need to know what happens in your lab in terms of maintaining temperature. This is an old paper from actually what were at the time our biggest competitors in Sydney, and this is looking what happens when you put an equilibrated dish onto different types of stage warmers. And the best results here was with a surface that was set at 37 degrees C and the lid is on the dish. But you can see that even at 37 degrees C, you are going to see cooling. And this cooling occurs because there's an air gap under the dishes, and we're going to explore that more later. The worst thing you can do, of course, is put it onto a heated stage with a hole in the middle. So in the old days, we used to use for ICSI, we used to use stages with just a heated stage with a hole in the middle. And these, even if you set the stage at 38, they cool down incredibly quickly. I mean, this is just a minute, and it's down below 34 degrees. So you need to know what your stage warmers do, what they achieve. So you need to measure it to find out what is happening. In this case, this is temperature regained. So dishes were pre-equilibrated at 35 degrees, and they put into a big box incubator. As you see, with big box incubators, they get even colder to start with for the first few minutes, because when you open the door of the incubator, the environment falls out. So you've got to warm up the incubator again as well. And it takes for almost forever, more than 20 minutes, before they're getting back anywhere near 37 degrees. And we found that in the very early days of the optimization work we did in the early 90s. And that's why we started, well, that's actually what led to the development of the tube warmer and to improved workstations. And you can see that the best equilibration is putting these dishes into, in this case, it's a mink incubator, and they get back to 37 degrees C in about 5 or 6 minutes. So, important thing is, we really don't want to get 35 anyway. So if we got them 37 degrees and put them in a mink, they will stay there. So it is an incredibly stable platform. And the reason this difference occurs is because in box incubators, we have this air gap between the bottom of the dish and the shelf. And the shelf itself is a thin sheet of metal with holes in it to allow the air-gas mixture to circulate through. So the heating of the dishes, or the contents of the dishes, the culture medium, is actually quite indirect. Not much comes from the shelf. There's an air gap to insulators there, so it's a slow transfer of heat. So that when we designed the mink, we decided we wanted a solid state incubator with a large thermal mass, actively heated, and designed so that the dishes sat on top in direct contact with that heated metal block. So there's a large amount of heat available. They will equilibrate very, very quickly and be very stable, because the large metal block will keep the temperature stable. So this is what led us to develop the mink, and later on the BT37 when we were working with planar. So this air gap has been known for quite a long time. This is some work done by Anders Bech Pedersen at Nordic Cell. In this case, looking at the issue of the dish with an air gap, you've got a heated surface, but you aren't getting the heat down into the dish. So for his handling procedures, they designed these thermocoins. It's a small disc of aluminium which fits underneath the dish, inside the outer rim of the dish, and it allows heat to transfer directly. So in this case, this is a dish at 37 degrees, equilibrated on a 37 degree stage with a thermocoin in place. And you've got about 5 minutes before it still cools down a little bit, because the dish being open will still lose some heat. But if you didn't have the thermocoin present, it's down to 29 degrees after 5 minutes. Even if the surface is at 37, the dish is at 37, it cools down very quickly. Things cool much faster than you might imagine. So you need to do experiments to see what happens in your workstations. Whatever your configuration is, find out. And these sort of carriers that you see in some incubators, do they do what you think? Do they actually improve temperature control? So think about the physics. So this is a dish inside one of these carriers, and note that the carrier really does not contact the dish in any meaningful way at all. So it's not going to transfer heat into the dish. It might feel nice and warm in your hand, but it's not going to really heat the dish. And what's worse now is the air gap that we had under the dish is now exacerbated by the thickness of the metal that is contributed by the metal carrier. So it does not work. These things do not work for carrying dishes to keep them warm. So don't be deluded into thinking this will improve your temperature control. This is actually the rapid recovery system from Shivani, I'm sure you all know. And in this case, what happened was that they took these carriers and then had them engage into a metal plate that was actually looking like the inside of a mink incubator or BT37, for example. And this actually means that the dishes sit in direct contact with a large metal mass, which If it's inside an incubator, it's heated by the incubator. If it's on a heated surface, it will occur with the heated surface. But of course, you still need to know exactly what happens by making measurements in situ. Don't just read a brochure, listen to a sales rep. Find out, test it yourself, make measurements. So we know we need to keep the follicular aspirates above 35 degrees because once you get below 35, the spindle, the magnetic spindle starts to depolymerize. And of course, when it warms up again, it will repolymerize. But I'm going to show you some experimental work from various people that show that it takes longer to repolymerize than it does to depolymerize. So if you cool the oocyte, you risk aneuploidy because when the spindle repolymerizes, it has to reconnect with every chromosome. And if it doesn't have enough time to reconnect properly, then of course, when you get the fertilization occurring and that first division, you risk aneuploidy. So oocyte causes cooling. Oocyte cooling causes spindle depolymerization. And if it happens a number of times, it will just add up more and more because it takes longer to repolymerize and depolymerize. And temperatures fall very, very quickly. You've got only a few seconds before a dish has gone from 37 to below 35. So again, you need to know what happens in your laboratory because it depends on the temperature in your laboratory, the amount of airflow, and the way you actually do things. So temperature control is very important, particularly during follicular aspiration. No one has yet come up with a way of keeping the thin tubing warm between the needle and the tubes, but this original tube warmer was developed as a result of our work at Sydney IVF and has led to a number of other products. I'm aware that Siobhan has got a newer model for their tube warmer, but other people got tube warmers. There's quite a few of them around now, but these came about in the very early 1990s. To allow us to keep the aspirates as warm as possible, while keep the tubes warm while the tube is filling with the aspirate, because the Nurse Hand Mark 1 tube warmer does not keep tubes at 37 degrees. So we need to control the temperature during follicular aspiration, during transport of aspirates to the workstation, during the egg search and the handling of the cumulus oocyte complexes, and of course during the IVF insemination or during the ICSI procedure itself. And along the way, we've got to think about osmotic stress, because when we get evaporative loss of water from the follicular fluid or culture media, the osmolarity will increase. And we also protect about pH shifts. I'm not going to do a lot about pH shifts, but we have to think about that as well. So we've got to think about all of the physical chemical things that will happen all the way through the procedure, looking at temperature, pH, osmotic stress, everything. So the short bit about bicarbonate buffers is that we know that bicarbonate buffer requires the correct amount of CO2 in the atmosphere above the culture dish to allow that CO2 to dissolve in the water to an appropriate concentration so that with the water it makes carbonic acid and equilibrates with bicarbonate. And we know how much CO2 we need because it's dependent on the amount of bicarbonate, the temperature, and the atmospheric pressure. Now, why the atmospheric pressure? Because it's not the percent CO2, this is the partial pressure of CO2. We need to maintain for 25 milliequivalents per liter of bicarbonate, which is as normal in most culture media, at 37 degrees at sea level, we need 44 millimeters of mercury partial pressure of CO2. If we go up in altitude, we need to increase the percent CO2 to maintain the same numbers of millimeters of mercury partial pressure. This is some old work, very impressive work done by Debbie Blake, and was presented at the Alpha meeting literally 25 years ago. And in this case, she'd equilibrated dishes and different types of dishes, 5 mils in a 60 ml dish with either gas or ungassed oil, that's the green and blue. But the one that matches most common practice today is the 50 microliter drops under oil. It made no difference whether the oil was gassed or ungassed, but they take a long time to equilibrate, as we know. And she had thermocouples in the dish, and she took them out of the incubator. And after 3 minutes in there, in fact, within 2 minutes, the pH was above 7.45 in these dishes, all the dishes. So having a lot of oil or a little bit of oil does not really protect the outgassing. The bad news is that if you've got a lot of oil and small media droplets, it actually takes longer to re-equilibrate, so to regas, than it would if you had a larger volume of culture medium. That may be counterintuitive, but it's about getting the CO2 through the oil. And the thickness of oil when you've got droplets is much thicker than it is when it's just a float on top of a large volume of culture medium. So just remember, for 50 micron droplets under oil, regassing takes about 20 times longer than the degassing. So it's a massive risk of pH instability. It takes 2 minutes to deequilibrate and 35 minutes or so to reequilibrate. That means that if you're trying to use these sort of gas funnel things to put some nice premixed gas around the dish, it doesn't do any good. Brief periods under this gas funnel cannot achieve regassing because it takes a long time. So the benefit is minimal. So the pH will rise, embryos will be stressed. And this happens when the out time, according to David Blake's work, is 2 minutes, less than 2 minutes. So if you keep putting the dish back under one of these funnels, it doesn't do any good because it takes 20 times longer to regas than degas. So basically the maximum amount of time that a dish should be exposed to air is less than 2 minutes. And that's what we try and stick to in any of our procedures. And in fact, the way we design labs and where we design our SOPs, a dish is out of the incubator for a few seconds, and that's all. So with the lid on, that makes it incredibly stable conditions. So we know from old work, this is from 1987, Sue Pickering and Martin Johnson, that if you look at spindles and oocytes, obviously from the mouse, that if you put the spindle, put the oocytes at 25 degrees C, then over time you will get a decrease in the number of spindles you can see. If it's 18, it happens much faster than 25 degrees. After you've taken these eggs and put them back to 37, half of them have got their spindles back after half an hour, and it takes about an hour to get all the spindles back. So it takes a long time to regain all the spindles, even though it's a relatively slow procedure to lose them. But you still don't have many minutes at 25, but we hope no one ever put eggs at 25 degrees. And in case of chilling, here's some studies on human metaphase to oocytes as part of some research related to oocyte freezing. You can see that it doesn't take very long, 3 minutes, 4, 5, 6, for the spindles to completely break down. So we know that we don't have very long at all when we're cooling for the spindles to disappear. So spindles disappear, and of course they will reappear. So this is a more recent study, and this is again a mouse for the experimentation. But after vitrification and just after warming, the spindles that you could see nice and clearly before you froze them or vitrified them have gone. And then whether you have just left them in the medium for 2 hours or you've got the vitrification 2 hours after warming, you get the spindles reforming very nicely. So the spindles will reform. The problem is you must make sure you've left enough time for all the chromosomes, seen in blue here, to be properly reconnected to the spindle. Otherwise, you still have the risk of aneuploidy. So another study looking at some clinical impacts on freezing or vitrification of oocytes. Yes, we know upon cooling the spindles, tubulin depolymerizes. It repolymerizes faster after vitrification than it did after slow freezing. So vitrification is one of the reasons I think that vitrification is more successful in slow freezing. This means that spindle recuperation is complete by 2 hours post-warming rather than about 3 hours post-thawing. So when you're going to warm your eggs after vitrification and you're going to do ICSI on them, you must make sure you wait long enough for the spindle recovery to be complete. Otherwise, you will certainly run the risk of aneuploid embryos. And we know that during the ICSI procedure itself, if you maintain temperature optimally, you will see more spindles, and you will obviously get better fertilization rate. And this is an interesting study from more than 20 years ago. There's two systems. It doesn't matter what they are, they're both still around in one way or another. And you can see that system B is obviously much better at maintaining spindles. And this actually means, in fact, you get a higher fertilization rate at ICSI, and the embryos are much more functional. So we know that we need to maintain the temperature during the ICSI procedure. So you must know that what's happening on your ICSI rig, that the eggs are not being allowed to cool down on the ICSI rig. You don't necessarily need a pulsoscope to see it for evidence. You just know that you need to keep them warm. If they get below 35, you've got a problem. So keep them as close to 37 as possible. So is this cooling a significant problem? Well, yes, it is, because you've got this issue now where from Santi Monet, you can see that the euploidy rate in donor oocytes can range between 40 and 82%. So labs up here are doing well, labs down here are terrible. Doesn't matter where they are, what they say. donor oocytes in these labs only have half the success rate as donor oocytes in these labs. And we got indication of this with some work we did at Santi back in the late 90s, where we looked at cleavage stage embryos and looked at the mosaicism rate amongst the individual blastomeres of those embryos after they'd been fished. So we know that cooling is a significant issue, and I suspect that the cooling of the oocytes or cooling of eggs during ICSI or the time that the ICSI occurred after the oocytes were warmed will all be impacting euploidy rates. So you've got to understand that everything you do can have negative impacts. So thinking back to the early days of egg search processes, this is actually a picture from the Born Hall book. This is one of the early labs in Vienna. This is the workstation used in France where the first French IVF baby was made. And you can see that these are just warm stages, but even back here, this was a modified baby incubator. And I first saw this in the lab in Paris in 1981. So these sorts of workstations are not new. We know there's many permutations with built-in heated surfaces, big ones, small ones. Even if you use this as a small laminar flow cabinet sitting on a bench with a heated stage on a stereotype microscope, this will work reasonably well, but you still need to know exactly what's happening inside the dish. And in fact, this is a system that was in use in one location when Sharon took over the lab and replaced it with one of these IVF chambers. And the tube warmer was already in place. But basically, you can see that changing from laminar flow type workstation to an IVF chamber workstation increased the number of good 6 to 8 cell embryos on day 3 by 50%. Now, this is about very old work. This is showing that we've known this for a very long time. This was presented 23 years ago. So we've known this for a long time, and yet people don't pay attention to it still. These workstations look very nice, and they are vertical laminar flow cabinet quality, air up to ISO class 5, nice heated work surface, but you need to equilibrate them properly to make sure that you maintain the dish contents at 37. And often these surfaces are only set at 37 degrees, so we know that they will cool down with the lid off the dishes. So you've got to think what's happening, and the answer is it's windy in there. You've got airflow, which is at ambient temperature, high volume and velocity, and this creates substantial direct cooling as well as evaporative cooling. So you've got direct cooling and evaporative cooling. It's not good. And of course, it's very difficult to maintain the PCO2 in the work area, so you've got pH instability as well. So are these really fit for purpose? Massive numbers of labs around the world use these sorts of workstations because everybody uses them. Are they the best workstations? I have never bought one of these workstations for egg retrieval, not in 33, 34 years. When you're calibrating a surface to keep the dish contents or the culture drops at 37 degrees, you need to remember that the source of heat is the metal surface around the observation area, which is that glass plate. And the glass plates are only heated by conduction from the sides, from the metal that's around them. So the glass plates are cooler than the metal surface around them. And of course, you've got heated metal surface holes. They're very bad, and we will ignore those because I don't think everybody still uses them. At least I hope not. So we need to calibrate our workstation to keep the temperature in the metal hot enough to keep the glass hot enough so that the dish is kept hot enough to keep the medium in the dish at 37 degrees. And you can't guess that. You have to do it and measure it and calibrate it. Now, the issue, of course, comes if you're going to use different types of dishes, which have got different surface areas of exposure, particularly if there's no oil float, and also with different thickness of air gaps. You need to calibrate appropriately to make sure you've got the correct temperature in the dish. And people say, well, we don't, when we're using our cabinet with a dish like for egg retrieval or whatever, we don't use the air, we turn it off. So because we know it cools it. Well, if it's off, what's the point of using a workstation like that? Because the only purpose of those workstations is to create a significant airflow of ISO class 5 air. And the dish lid on or off, remember this. With the lid off and no oil, as in during the egg search, the evaporative cooling effect increases as a function of the square of the dish radius. Air equals pi r squared. So if you've got a 60 millimeter dish, you've got about 28 square centimeters of surface area. 90 millimeter is more than double. 100 millimeters is even larger. So if you're going to use big dishes, understand that you will get much more cooling. Even in a 37-degree workstation, unless it's completely humidified, you will still get a small amount of cooling, and you'll also get osmotic changes. So we know that cabinet airflow causes evaporation during dish prep as well. So this isn't just restricted to what happens during egg retrieval, it happens during dish prep. And Jason Swain's published a nice paper on looking at how you do your dish prep to reduce the impact of this on the osmotic concentration of the culture medium. Optimizing egg retrievals. Well, this is results from a clinic which I'm allowed to show because we're very happy with there's actually an audit. We call it an operational performance review because it's just a slightly less threatening expression for the lab people before they get there and they get to know us and that we're not going to bite. But in this case, they were quite happy with their results. It's about 37% clinical pregnancy rate per embryo transfer across all patients. And it varied around plus or minus 10% per month within that range. And some simple procedural changes using a tube warmer to keep the follicular aspirates warm, make sure the LAF cabinet surface was maintained 37 degrees in the dish during egg search, and also avoid exposing the cumulus oocyte complexes to cooling during handling, so not holding them in the fine glass pipette for longer than necessary. So pick up an egg, move it. Pick up an egg, move it. Don't pick up an egg, pick up an egg, pick up an egg, move them. By those simple changes, the control mean increased by 30%, and the variability reduced from 10% to plus or minus 5%. So we have a substantial improvement for some very simple changes. So I've been talking about these workstations. I said they're not new because that first one was in Paris in the very early days of IVF. And they originally were modified neonatal isolates, and everybody had to stand up while they worked at them, like this IVF chamber from Hoffman. They were temperature and CO2 controlled and humidified. Early models used unfiltered room air and just added CO2 to it. Later models added HEPA filtration and even VOC elimination at this generation. Later models were also height adjustable, and you could stand at them or sit down and work at them. And the latest model that was shown here actually has a dual microscope system, which is a digital inverted microscope as well for doing fertilization checks and detailed embryo assessments. But these workstations come from many people. They've been around for a long time. We've used this particular product range, which is from an Australian company, because we helped improve these in the early days. We contributed to the design of this one and also to the design of the latest generation. And this latest generation tech event was an Australian company. It's now owned by Planer, and their temperature, humidity, CO2 controlled, near class 5 air, built-in photocatalytic VOC removal. So it's a very good workstation. There's no opportunity for off-gassing of CO2 from the dishes, so culture medium pH is maintained throughout the handling and assessments. You can even get a low oxygen module that you can put in the side here if you want to just hold dishes in there under more incubator-like conditions. Height control is motorized with memories, and you can even get an ICSI workstation built into the same sort of cabinet. So this is the sort of workstations that we want to use to effectively eliminate many of the variables in temperature control and pH control and osmotic control for the dishes. So the thing to remember, though, is that even with these workstations, and this is small cases but also has humidity, even with humidity control operating, since it's usually only maximum 60 to 70%, and it can take quite a while to reach 70%, there is still going to be some evaporation from the dishes. So there still is some risk of follicular fluid cooling during prolonged exposure of open dishes. So if you've got dishes open, even in one of these workstations, you will get a little bit of cooling. So that's why the environment is set a little bit above 37 degrees to keep sure the dish contents stay at 37. But you have other workstations, Aztec, Unica, and these are dry. There's no humidity control, or if you use one of these with the humidity turned off, there will be significant evaporative cooling even at 37, and in fact, probably more at 37. So temperature control has been much more difficult in these sorts of workstations than these workstations. So don't just read a brochure, listen to a sales rep or whatever. Think about the physics. It's only high school physics, it's not complicated, it's not quantum physics. Just think about what's happening with temperature control in the workstations and make measurements. It's not expensive, it's not difficult. Just make measurements and know what's happening in your lab, in your workstations. We've talked about the oocyte, and we know that mature oocytes are what we're looking for, but we know we recognize immature oocytes. What is not recognized much these days is post-mature oocytes, but it is important because these post-mature oocytes can fertilize and they can make nice blastocysts, which can even implant, but they have a much lower clinical pregnancy rate than blastocysts derived from mature oocytes. So recognizing these means you do need to spend more time looking at the oocytes. I wish more people would do this because we used to do this routinely and it gave us a lot of useful information. But nowadays, if people aren't doing it, they're missing the whole point about post-mature oocytes. And you can't really do this in a non-humidified workstation because you have too much risk to the oocyte. So when we're assessing oocytes, the Istanbul consensus says a good COC has expanded cumulus radiating corona. Other observations should be noted as exceptions because people can't agree on anything. Now, since then, we get some AI things that say they can recognize good, bad oocytes. We've been using a grading scheme literally for 30 plus years. This comes from a paper published from Calgary, but this paper was published a few years after Maha left. But basically, this grading scheme was developed in Calgary. It was elaborated during the 1990s when I was at Sydney IVF, and we've been using it ever since with labs that will listen to us over the last 20 odd years. And it gives you a good way of grading an oocyte, getting a score out of 20. Is it useful? Well, and when an AI company says that they have got the first thing that ever tells you anything good about the eggs, they're wrong. They just don't bother reading old papers. It also spoils their sales story, I suppose. But in this case, we've grouped the oocytes by the score, the good ones, the fair ones, the poor ones. And it's young patients, reasonable stimulations, only ejaculated sperm. Fertilization rate in IVF and in ICSI is associated with the good oocytes and is not as good in the fair oocytes and worse in the poor oocytes. But it's not just fertilization, it's utilization rate. This is the proportion of embryos that were deemed good enough for transfer or cryopreservation. And we can see that the good oocytes produce more embryos that are going to be usable. Fair ones, fewer. Poor ones, even less. So this simple grading scheme can be very informative in telling you what you can judge about the oocytes at retrieval. But you cannot do this in a laminar flow cabinet workstation. Not safely, because you'll get prolonged exposure to the circulating air, and you'll get more cooling, and you'll get more osmotic shift and pH shift in the culture medium. Post-mature oocytes, just as a last thing, are a problem, but they're not well recognized, as I said, these days. And these are results from an audit we did from highly regarded, very successful ART center we worked with for quite a number of years. And this is following a perceived slight, about a 5% reduction in their clinical pregnancy rate. So we went in, did a repeat audit. The lab procedure KPIs were all found to be consistent for the lab, but the oocytes were darker, more granular, and the lab was not really very happy with them. So there's nothing we could see in the lab to explain any changes. So we reviewed 100 stimulation cycles, compared the actual trigger day to what was the protocol-defined trigger day. And 75% of the cycles were triggered late. Not just a day late, often 2 days late, even 3 or more days late. And when you have delayed triggering, yes, you get more M2 oocytes, but also the likelihood of post-mature oocytes increases, and you have this risk of elevated progesterone trigger day. So measuring progesterone on trigger day can be very informative as an indicator of the increased risk of post-mature oocytes. You've got to beware post-mature oocytes. I said, although few embryologists are trained to recognize them, I think this would be a very important KPI to reintroduce somehow, because it will tell us whether the problems we're seeing in the lab are due to things in the lab culture systems, or going right back to when the physician decided to trigger the retrieval. Stimulation information is very important in troubleshooting, and many of the oocyte quality issues, such as ooplasmic immaturity, often underlie perceived lab problems. They say they're lab problems, but they're more likely due to oocyte quality issues. And key information needed for understanding the adequacy of stimulation, especially the appropriateness of trigger timing, is essential for troubleshooting. And it should be integral to proper cycle reviews to improve best practice and quality improvement in the clinics. And without this sort of information, we can't troubleshoot. We can't go in and say, is this caused by oocyte cooling during handling, egg retrieval, whatever, because the eggs could be already compromised before they even get into the lab. So without knowing all this information, we can't really troubleshoot issues in the lab that may or may not be due to oocyte cooling. We call this troubleshooting in the dark. And the appropriate importance of cycle reviews. This is what happened in one clinic we worked with for quite a number of years. And you can see this is the blastocyst development rate on day 5. And over a 2-year period, it went up by about almost 1% per month. No changes in any lab procedures or staff. And all that happened during this period was a systematic review routinely of all treatment cycles by a multidisciplinary team. So physicians, lab manager, me, lab director, nurses, even the pharmacist came along through a 3-year period. This is the second and third years of this period. And the clinical staff was the same and the lab staff. But because everybody talked and discussed things, discussed the cycles, we understood a lot more about what was going on. And this is the end result. We got a lot more blastocysts. Of course, we had to recalculate our control ranges at this point, but it was I left the old control range in place just to show where we improved systematically across that period of time. So communication between physicians and labs will lead to better outcomes for sure. So take-home messages. Simple mantra: think like an oocyte. But as embryologists, you need to know and understand what the biology needs, and hence what the chemistry and the physics must be. Then you can use that knowledge to design and engineer your lab facilities to eliminate, or at least minimize as far as possible, all the conditions or factors that might adversely affect the gametes or embryos. You can use the information to engineer and then calibrate and maintain carefully selected equipment that is optimally fit for purpose to maintain the correct biophysical and biochemical conditions for each step of the process. You can elaborate and refine culture media formulations optimized for both gametes and embryos, human, not mouse. And this is part of product design and product selection. You can use the information to define the requirements, the proper and safe handling of gametes and embryos. This is process design and process optimization. This is where we use our FMEAs, those failure modes and effects analysis, proactive risk management, and quality control. And we must, of course, respect the timeline in all this. And then, having done all this, we can monitor each process using our indicators, our KPIs, and relate them to benchmarks. And this is where we get our quality assurance and our quality improvement process going on. So think like an oocyte. Remember you're a scientist and not a technician, and that you are going to understand all this stuff. And when you're training new people, teach them this stuff. Don't just teach them what to do, teach them why you want them to do this way and why you don't want them to do things certain other ways. Thank you very much.
Thank you, David, for such an enlightening and comprehensive webinar, this talk on, and your message, take-home message. I really like the message that think from the angle of an oocyte, and then only you will be able to achieve all the parameters that are required to maintain the right osmolality, the right pH, the right temperature, everything. Then only we will be able to achieve the all these parameters.
Remember, you have to think like a sperm or think like an embryo at other appropriate times as well.
Right. So whatever condition your gametes or embryos they are in the incubators, so you have to make up your mind to think from that angle. That's very true. And David, we all know that the highest degree of danger is to the spindle. That is more detrimental spindle. If there is any spindle shift that will lead to either aneuploidies or it will lead to. So what is the basic thing that how can a clinic know that there is some shift in the spindle or there is something wrong with the spindle? So is there any parameter?
Unless you want to examine every oocyte at every throughout the process using a pulse scope or a spindle view, which is an expensive way of checking things. You just need to know you maintain the temperatures. I mean, if the spindle is a problem inside the egg anyway, it is an issue that you can't fix. If there's spindle abnormalities, then they are inherent to the egg for whatever reason. But the important thing here is we're trying to avoid creating any iatrogenic, doctor-caused, lab-caused problems. And that's simply done by keeping the eggs warm. You should maintain them as close to 37 as possible. Our process for us would be considered to be poor if the eggs ever got below 36. And I say that's why we have not used a laminar flow cabinet for egg retrievals in any lab that we've been responsible for, or where we've recommended the procedures to people. We've never used a laminar flow cabinet in since 1990, 91. When I moved to Sydney IVF, we just changed. I knew about the one in France, but we didn't have access to them easily through the 80s. Because there was no commercial manufacturer. You had to have someone who was going to tinker and buy a unit and rebuild it for you. But 1991 was when we realized the importance of this, and we stopped using anything other than those controlled environment workstations. Temperature controlled, CO2 controlled, humidified as far as you can get it. But you can't get it to as high a humidity as you might like because it becomes condensing and makes a mess. And it's also very hard to achieve that. But you minimize as far as possible. You control things as best you can within the general conditions. And you can't have a warm, humidified workstation inside a lab that gets cold at night because it condenses. So you have to, this is why as we've learned more about HVAC systems and air quality in labs, you need to maintain the temperature of the lab all through the day and the night. It might seem it's not very environmentally friendly, but you need to do that to help maintain the conditions for everything that's happening. It's just part of the cost of doing IVF.
Right, that's a part of it. Yes, can't just skip that. And with this, we open this forum for any questions. So you can just write in the chat box if you have any questions, and we'll start taking the questions. And with this, I can see the first question. That's very interesting question. In fact, from Nibhash Kumar. So he says that, do you want me to read out the question for you, or
yes, please, because I'm not sure which one.
Sure.
I'd be more focused on the chat box as I was about answering questions then. You can moderate.
Right. Sure. So, sir, as we know, temperature fluctuations can cause spindle problems, and depolymerization can increase the rate of aneuploidy. My question is, why do we perform vitrification at room temperature? Does this mean that vitrified embryos or oocytes, they have a higher chance of aneuploidy?
Well, you do the vitrification at room temperature because of the toxicity of the high concentration of cryoprotectants. So cryoprotectants are all to some extent toxic, but toxicity of these sorts of chemicals is much higher at 37 compared to room temperature. So you avoid some of the toxicity effects of those high concentrations by doing it at room temperature. But yes, we know that spindles will depolymerize during the process. But we also know, and I showed some nice images from other people's research, that shows that the spindles will repolymerize afterwards and will reform. You just have to make sure you leave it long enough. And it says it's probably at least 2 hours to be sure of getting the spindles to reform in warmed vitrified oocytes. So you need to leave them that long before you start trying to do an ICSI on them. So there is a risk. I'm not sure that there's, I'm not aware of any evidence that suggests that warmed vitrified oocytes have a different aneuploidy rate to slow frozen ones. Probably because the slow frozen ones it would seem to take even longer to repolymerize the spindles and reconnect with other chromosomes. So part of the problem with slow freezing is likely that the eggs were being inseminated or doing ICSIs before full spindle recovery had occurred and reconnected with the chromosomes. So it's not just spindle reforming, it has to reform and reconnect with all the centromeres. So you just got to make sure you leave enough time for it. Can't be in a rush. You've got to follow the timeline that's determined by the biology. And remember, slow freezing also cooled things down. We know the spindles always depolymerize during those processes. We knew that. It was a question of designing your processes to make sure that you allowed the time for the spindles to reform and reconnect with the centromeres.
Exactly what happens, like when we say that the spindle it gets disturbed, so exactly what happens, the spindle what
Well,
are the cascade of events?
It's quite simple. The spindle is made up of microtubules, and the microtubules are made of tubulin, and tubulin automatically polymerizes and makes the microtubules.
Yeah.
And when it gets cooler, the tubulin molecules depolymerize. So the microtubules just disappear.
Exactly.
And as they disappear, they leave the chromosomes stranded on the metaphase plate in this case. And so they're just sitting there. And when it warms up, the spindle will reform. Of course, it'll be reforming around the two microtubule organizing centers, which come from the centrioles, and they will reform. And as they reform, they will reconnect with the centromeres of the chromosomes. But if a chromosome has drifted away, it could be lost, so that aneuploidy, or they may not fully reconnect. And when the spindle initiate fertilization, and the two MTOCs have organized the microtubules to move apart and separate the two sister chromatids, you end up with a chromosome, two copies of one chromosome going one direction, for example.
Wrong direction.
Yeah. So that's the issue. Just, we know it will fix itself. Give it sufficient time.
Yeah. So exactly, that's why many of my students, they often ask me this question, that what exactly happens to this spindle? And I thought that who's the best that you to answer this?
Well, if you have access to a if you have access to a pulsescope or a spindle view, let them watch the mouse eggs just losing their spindles.
Maybe they can watch the videos on, maybe if they are available on YouTube, if some videos are there, so they can watch the videos on YouTube.
Exactly.
They'll get to know exactly what happens. So the next question, that's from Imran Khan, and he's asking that in some cases we observed polar body disappearance after warming of oocytes. Is this a normal phenomenon or something related to vitrification warming protocol?
I don't know for sure, because it can't be a normal process, because vitrification is not a normal part of biology. So it is something related to what you've done with the vitrification, the warming. Polar bodies are just junk anyway, so they have no significance unless you want to be doing polar body biopsy. So even when you're looking at fertilization, doing fertilization checks, you will sometimes see that the first polar body, by the time you're doing a FERT check, has fragmented. So you've got the second polar body, the first one, one of them is fragmenting. The polar bodies break down and disappear anyway, so I wouldn't be worrying about them in any significant way.
Okay, the only problem is, I think, with the ICSI, during the time of ICSI, you will exactly won't know that where the polar body was and which way to go the injecting the oocyte.
That is true, but if you'd have to lose. Now, it's true, but at the same time, there are now people talking about doing automating ICSI without doing reorientation or proper orientation of the oocytes. So, I don't know. Someone would have to do some very careful experiments not with human eggs ideally, but probably someone will somewhere, just to know how much of an issue that really is. But the amount of distortion that's occurring in the oocytes, and with some of the more exotic ICSI techniques, and some things that happen with non-piezo ICSI, where you're getting these huge funnels, injection funnels cause, those are massive disruptions to the oocyte already.
Okay.
So I don't know. I mean, yeah, we want to avoid hitting the spindle, but at the same time, That's a presumption. I'm not sure of any published evidence to show that it does have the same effect. I wouldn't be wanting to go and do it, but at the same time, I would suggest that people still make sure they orient them as best they can. If they lose the polar body, then yes, it is a loss. If that's a big issue in your lab, then that's when you should be getting a spindle view and being able to make sure you orient the eggs correctly before you do the ICSI.
Right, right.
And that may be a reason for people getting more spindle views because, or oocytes, whatever you want to call them, simply because they're able to make sure these warmed vitrified oocytes that no longer have a polar body for the correct orientation can be oriented correctly by actually visualizing the spindle.
Yeah. So, but David, we had a spindle viewer for trial at our IVF setup. So, but what I observed, in my observation, spindle view it would take longer duration, longer time for ICSI,
Yep.
and the net result was not that encouraging. So, but yes, you get to know the exact the view. You could see the beautiful spindle.
I've never seen a need for it in routine ICSI, except perhaps if you do have this problem where you lose your marker of the polar body to orient the oocyte correctly, then that may be a situation where it would be beneficial. So someone needs to do the study and find out.
Okay, okay. With this, I would like to introduce my co-moderator for the day, Priyata Das. She is the clinical embryologist and a trainer at Indra Fertility Academy. And
morning,
she was
Priyata.
the one who was
Good afternoon.
communicating to you all the time. Priyata, maybe we can take next question then.
So first of all, I would like to say that it was a delightful session today. Such an interesting topic, and I was really looking forward to hear you speak again. Before we move on to the other questions, I would like to ask you one question. We were talking about you were talking about maintaining the temperature during ovum pickup, right? So
Yep.
would you suggest preheating the tubes as well before we start pickup, so that when we collect the follicular fluid, it is already in a tube that's already warmed at 37? Or is it okay with just putting them in test tube blocks that have been warmed already? Which one would you prefer?
We have pre-warmed the tubes forever. I can't think when we first started doing that. So when we setting up a facility, we would put a large dry bath incubator, which has all the blocks with the holes in them, and we would put the unused tubes for use in that blockage to make the tubes warm. We would have the tube warmer, which is a separate smaller unit, which is designed to go into the clinical field, into the sterile field, and that tubes are going to there from that dry block. They would go into the tube warmer, and then the aspirate would be collected into the tube. And that tube is then transferred to the laboratory. The tubes are therefore pre-warmed in that big dry bath. And in fact, there's probably even more tubes kept warm in an incubator somewhere nearby as well. So the tubes have always been pre-warmed. What is an interesting question, though, is some labs will actually put 1 ml of Heapy's buffered or MOPS buffered medium in the bottom of the tubes when they're equilibrating them. And so long as you make sure you put it in at the appropriate time to allow adequate time for it to warm up to 37 degrees, then that could be beneficial, because when the egg is coming down that tubing from the end of the needle, it flows very quickly. So it will cool down a bit. We don't know exactly how much, but it will cool down a little bit going down there. And it obviously can't be that much of a problem, because that's the typical process. The question, though, is if you stop aspirating, there is always some fluid left in that long piece of tubing, and that will cool down very, very, very quickly, because it's a thin piece of tubing in a room that is usually about 20, 21 degrees with a large amount of airflow. So when that fluid goes into the tube, then would having some pre-warm medium have helped. Some people do it, most don't. No one that I'm aware of has actually done a formal, proper prospective study to see if it makes a difference. Something that could be useful. It could even be done initially just by measuring temperatures and looking at what happens having got a thermocouple inside the bottom of the tube and measuring the temperature of the fluid as it arrives and how it takes to warm up, things like that. So we don't have to do something involving eggs to start with. I mean, the reason we invented the tube warmer was because we did dummy procedures. We actually saved follicular fluid and we put it in a flask in a 37-degree water bath. I put the water bath on the procedure room table where the woman or her bottom would be. And then we did dummy procedure. We aspirated that fluid through needles, tubes, intercollection tubes, took them into the lab, into our workstation, poured them in a dish, left it for the appropriate amount of time with appropriate manipulations to match an egg retrieval, egg search, and then put it into a big box incubator. And we realized that they were down about 29 degrees, and it took about 20 minutes for it to warm up again. Now, we realized that this was completely useless. So that was why the original tube warmer was developed with Cook, to keep those aspirates warm. So, gives you more experiments to do.
Okay, so this was an interesting one that you said about keeping pre-equilibrated media in the tubes beforehand.
Yeah.
That is something we could try and see what happens. Thank you for that answer.
Yeah.
Now moving on to the next question. Dr. Ravi has asked, I need some clarification. What is your opinion on gametes or embryos thawing at room temperature and warm plate? Because some papers show that warming at 37 degrees Celsius gives better success rates.
Absolutely. This is a classic one. So when you've got a frozen, let's say it's frozen as opposed to vitrified. Vitrified, you know you have to bring them back incredibly quickly. It is the most crucial part. The rate of cooling in vitrification is less important than the rate of warming after vitrification. But we're talking about freezing now. So when you've got a sample that's a specimen that's frozen, -196, it's been frozen, so there's only ice formation in the extracellular part of the material. Inside the cells, you've replaced the water with permeating cryoprotectants, and you have got no ice. Problem is that as you're warming up, ice crystals will reform within that frozen state, will grow, and they will grow everywhere between about -132, which is the ice transition point of water, and the melting point of the solution, which is probably about -6, -8. So all through that time period, you've got ice crystals growing in the extracellular compartment. They're just getting bigger and bigger and bigger. And there's some lovely pictures from a guy called Merriman, and the work was as old as I am, and it showed very clearly that these ice crystals will grow very quickly, and they'll be micron-sized crystals that can destroy cells. So you need to get through that transition point as quickly as possible. So you need to get from -196 to the melting point as quickly as possible, which means you've got to put heat in as quickly as possible. And you will put heat in fastest if the environment exposing it to is hotter. So you might expose it at 37, but remember at close to 37, the permeating cryoprotectants become more toxic. So we always, I mean, for as long as, I mean, I think pretty much ever since we first started doing embryo freezing, we would thaw the embryos using a water bath, because the amount of heat you can put into a specimen from air is much, much, much less than the heat you can apply via water, culture medium, whatever, by water. So a water bath. So you take the specimen out and you put it straight to a water bath. And you don't just put it in the water bath, you put it in and you move it around. Because if you just put it in and leave it in one place, then heat will transfer in from the water bath through the walls of the container into the specimen. But then you'll create a boundary zone around the outside, which is where the heat's been depleted. So the rest of the water bath is at whatever temperature, and it's cooler to the specimen. It's like if you stand in a cold swimming pool. Eventually it feels warmer, and that's because you've generously been warming the swimming pool with heat from your body. This is just the reverse process. So to avoid the boundary zone, you move it around, and that will give you the fastest warming. Do you do it 37 or 25? We've always done 25 because we want to avoid that last little bit of risk of toxicity of cryoprotectants. And going up to 25 in water will give you the a fast thawing rate with minimal risk of the toxicity, or minimized risk of toxicity. And that's how we always thawed things. We do the same for sperm as oocytes and embryos, for slow frozen.
So it's the toxicity that hampers. At 37, we have more toxicity compared to the room temperature, and that is the basic fundamental why we want.
And the things you've got to get from the frozen state to the ambient temperature as quickly as possible. Ice crystals will grow as long as you spend between -132 and about -6. Ice crystals will be growing all the way through there, and this is why straws are so much better than cryovials. You can't get heat into the center of a cryovial very quickly at all. I published a paper in about 2004 on sperm cryobanking, but looking at cooling curves and warming curves inside containers of different diameters, and you cannot achieve the correct cooling rate in a cryovial. You certainly cannot achieve the correct warming rate in a cryovial. Straws are by far the better devices for freezing and thawing anything.
So, sir, as an addition to that answer that you have given and the question that was asked, so basically the warming kits that we have, they have three solutions, right? TS, DS, and WS. So what we usually do is we warm the TS at 37 and then we insert the straw in it so that we directly come from -196 to 37.
The device, the tip of the device.
Yeah, the device. So for DS and WS, does that also, those solutions also need to be kept at 37, or can a person, start thawing at 37 and TS and then move on to room temperature?
You follow the manufacturer's protocols.
Manufacturers.
So unless you're going to do some validation experiments and studies of your own, you follow the manufacturer's protocols.
Yeah, so I hope Mr. Ravi had gotten his answer, because he had reframed the question again. So that is why I wanted to clarify.
We'll jump on to the next question, I think, Priyata. So Mahesh is asking to refreeze some of the embryos. Can we move them to ES after DS step and VS? To refreeze some of the embryos, can we move them? Refreeze the embryo.
I'm not an expert in vitrification. You would have to ask what the kits are, what the products are, what the whole procedure is. That's not something you can give a simple generic answer to, and I would not want to make a simple statement on that.
Okay, and then the next is related to today's talk, that can we keep the COCs in dry incubator with oil overlay, or is humidity necessary?
Well, you tell me. It depends.
Exactly.
Dry incubators were never designed by embryologists. They were all designed by industry, wanting to put in integral gas mixers where you can't have humidity because it increases the risk of contamination. If you're going to put them in a dry incubator with an oil float, you need to make the measurements yourself and find out what happens to a dish without any embryos or eggs in it. What happens for the duration of time you're going to keep it in that incubator with the correct volumes of medium volume of oil, and measure the osmolarity of the culture medium.
Right. And you also explained in this slide as well that the oil, it takes a longer duration to equilibrate. So
for gassing it does, yes.
For gassing, yes.
Yeah. Yes. So the thicker, the deeper the oil, the longer it takes to regas. But you will need to know, if you're going to leave a dish in a dry incubator, you need to understand what's happening. You have to make the measurements. Check the pH. If you're measuring at pH, check the osmolarity of the culture medium to know it's correct. This is why modern culture media tend to be around about 10 to 15 milliosmoles less concentrated than the more traditional sequential culture media used in humidified incubators, because in many dry incubators you get a significant increase in culture medium osmolarity. And if you start off with ones that are a correct physiological osmolarity, they can be getting up close to 300 milliosmoles after 5, 6, 7 days of continuous culture, which is we'd never thought was a sensible thing to do. But they can end up being to osmolarity where we know it can start adversely affecting the embryos. So you need to know what's happening in your dishes, in your incubators, understand what's happening. And these are not complicated things to measure.
It's not complicated. What I can see, there are a couple of questions that from the vitrification itself. I think our audience is quite fascinated with the vitrification part and the warming part. So I won't be taking those questions. I'm so sorry, extremely. We will be just sticking to the today's topic.
Yeah.
And Preta, please see if you can
Yeah.
see any other question. Yeah, yeah,
Yes, sir.
tubes.
So,
Shubhangi.
so there's one more question. So Shubhangi has asked, how long do you recommend keeping oocytes in a buffered medium, say HIPPIS or MOPS buffered medium, post oocyte scanning, considering that the buffer is covered with oil?
Okay, there's a couple of things to think about here. We know that in the old days there was a lot of fear about these zwitterion buffers like HEPs, but that was more related to the fact that the purity of the buffer or the purity of the chemical was much less in the old days. People have done the whole process of IVF in mouse with HEPs present all the way through to some concentration, so it's not that toxic. The thing to remember though is that a HEPs buffered culture medium or MOPS still has bicarbonate, and in fact, it's probably about 10 millimoles of bicarbonate, which means that if you keep something in a HEPs buffered medium under air for a long period of time, that bicarbonate will disappear because the Henderson-Hasbach equation will not be in equilibrium. You will lose the bicarbonate as CO2 into the atmosphere above. So the bicarbonate will be lost. The pH won't change because of the buffering from the HEPs or MOPS. So you won't see that pH change, but you'll lose the bicarbonate. And that could be a problem because bicarbonate is essential for things like adenylate cyclase, whatever. So I would not hold them for longer than I needed to. I mean, it's a convenience, and you might use it, as I say, for doing ICSI. You might use it for an embryo transfer, but you don't just use it willy-nilly. You use it when you need to. You would use those sorts of buffers for freezing, vitrification, whatever, because you want to maintain the pH. In the old days, the early freezing solutions were actually made with phosphate buffered saline, which is cheap and easy, but in fact, it's something you should never have in an embryology lab because a pH of phosphate buffered saline changes as it cools because of the two salts. One of the salts is less soluble in cold water, so you get this precipitation of one of the salts, and therefore you get this pH shift. And in fact, when you freeze in PBS, you can end up with a pH of about 4 in the medium that's left around the embryos, and it's not so much in the freezer; it's when they're thaw, because the internal pH regulation processes are not functional for an hour or two after thawing, so you get this massive pH problem. So we worked that out in the about 97, 6, 7, something about then. So we obviously stopped using PBS at that point, but you can use these buffers as a convenience. I would still not leave them in there for very long. And remembering that you will still lose the bicarbonate, even though the pH will not shift because the zwitterion buffer is holding the pH. It's its job.
Okay, thank you, sir. There is one more question. Is there any consensus for oocyte grading? And if there is, then could you tell us which one is the best one to refer to?
Most people don't grade oocytes at all, full stop. They have a quick guess and say, oh, it's mature, it's immature, it's atretic. Yeah, you can tell if they're ugly, but that's not, as far as I'm concerned, that's not grading. The only grading scheme that I know of for oocytes that actually tells you useful information about the oocytes is the one I showed that we evolved in the 1980s, late 80s, and we've used in a lot of places ever since. It does tell you useful things about the oocyte. Problem is, some of the things we looked at and graded are not going to be the same because of antagonist protocols, because they change the appearance of the oocyte quite dramatically. We've all seen those very dark granular oocytes with the black floccular material in the perivitral space accumulating underneath the zona, for example. That changes how you appreciate the oocytes. And as I say, I would not be using that sort of approach, because one of the things we do is we actually tilt the dish to let the fluid run away from the cumulus oocyte complex. And if the corona cells pull away from the zona there at retrieval, then that indicates a risk of post-maturity, because the corona cells should still be attached with their transzona processes, so they should not pull away easily. So that grading scheme is there. It's not secret. And it does give you useful information about the oocytes. I'm not sure how good it's going to be in the era of antagonists. The AI systems that are around that are out. I'm not convinced by of their value, and I know a lot of people who've tried them and come back saying they're not very useful. It's just patients pay money for a decision which is probably the same decision they would have taken even if they hadn't paid the extra money. So to me, that's not useful.
The grading you had in your presentation that showed grade 1, 2, and 3, but they won't speak of that what type of abnormality that oocyte has, right? So they will just be graded as grade 1, 2, and 3.
Well, we've never used simple schemes like that. We stopped using schemes of that simplicity in the actually about the time we started doing embryo freezing. So that was in the 1980s. We've always used more complex grading schemes since then, because we want to know why an embryo or an oocyte is downgraded, not just it's being downgraded. We want to know why, and that's just our obsessions. We always like to have more information available than we might need, because if we want to try and do troubleshooting and we haven't got the information, we're troubleshooting in the dark.
Right. So, Preta, you can take next question.
I think this is the last question we have here. Can ovarian hypoxia cause sticky cumulus? And can we take any precautions there during denudation for those oocytes?
I have no idea why you might get the sticky cumulus with ovarian hypoxia. I don't know anything about that physiology, I'm afraid, so I can't answer that part of the question. If you've got sticky cumulus, then you would be denuding according to your protocols. I mean, how you perceive the sticky cumulus will vary by labs. Some labs will be looking at cumulus where they've got immature eggs. They've got post-mature eggs where the cumulus is almost gone. You've got to know what stage of the maturation the oocyte's in. But then there's also labs that will take the hyaluronidase and will dilute it. And they will say, oh, this one's not very good, but it's because it's perhaps less mature and you've diluted the hyaluronidase, it's going to take a long time to come out. Is it sticky? There's so many factors that can affect that sort of assessment or judgment that it's impossible to answer in isolation.
Right. So I think with this, we are over with the questions. And today, and once again, thank you, David, for the wonderful session we had. We really enjoyed listening to you, and it's always a delight listening to your sessions. So, okay, friends, so we'll meet next month on 3rd Friday with yet another webinar. Until then, bye-bye, good night, and have a safe weekend and happy weekend. Yeah.
Bye-bye.
Thank you, sir. It was great listening to you.
Very welcome. Bye-bye.
Bye-bye.
Bye, Naval.
Bye-bye. Thank you.